What's wrong with that? So thanks for inviting me. I'm really happy to be here again, and it's always a fun course. And so I'd like to welcome everybody to Montana. This is my favorite place in the whole world, over in Medicine, which is over in the southeast corner of Glacier National Park. That's Medicine Lake. So Gordon asked me to talk briefly about the biological station. So we're here right now at the Glacier Lake Biological Station. It's been around for over 100 years. It was one of the first places in the U.S. to do summer field courses. The first course was in 1901. The summer courses are still going on, and Gordon teaches one each summer. So Jack Stanford, who's been the director since 35 years or so, really is a big picture kind of guy, to quote Gordon. And so Jack's really trying to get a lot of people doing a lot of different things, focused largely on the lake, but looking from, starting at most of the word for it, genetics is what Gordon does here. And also largely limnology, lake ecology, but also looking at the cultural system. So there's a lot of research that's been going on here for a number of years since Jack has been director. And this is a slide that I think Gordon showed you. So we're here, and just north of here is Glacier National Park. And so here's the park, here's the biological station. So those mountains we're looking at, these mountains up here. And I just found out, I was excited to hear that Mike Miller is really excited about going to Triple Divide Peak. And Mike's going to be late entry. So Mike Schwartz and I have had an ongoing competition about who can get to Triple Divide Peak first so that we can urinate into 3 oceans at once. So this is Mike and I practicing this summer on a couple of double divides. We haven't been able to get to the Triple Divide Peak. So this is really, it really is the crown of the continent. So the water from here goes out to the Columbia Pacific. It goes down here to Missouri and the Gulf of Mexico. It also goes up here into Hudson Bay and the Arctic Ocean. So it really is, literally, it's the crown of the continent. And Gordon was talking about Stanton Lake. So the hike on Thursday, we'll drive up here and then down to Stanton Lake. And as Gordon said, there's grizzly bear sign there. Oh, sorry. Brief commercial announcement. So this is a wonderful book. So a friend of ours, John Ashley, published his book this year. So this guy, I'll show you the next slide. He's really crazy. He spends most of his nights taking pictures of Glacier National Park. So this is a book based on taking the pictures of Glacier National Park just after dark. It's on sale in many places around here. I really recommend it highly. This is just one of the pictures. So this is not far from here. So this is a picture that John took last December. So he hiked about 10 miles in December at minus 11 degrees Fahrenheit, minus 16 Celsius. And so this is a picture of Comet Lovejoy, and it was a lot of moonlight. And so this is just a picture of the trees that have a lot of moonlight. So it's really a fantastic book, and I really recommend it highly. Back commercial announcement over. So Stanton Lake. So it's in the Great Bear Wilderness. And this is a picture of a great bear. So there are grizzly bears around here. So I don't think Gordon's lost any students yet being eaten by grizzly bears. Have you, Gordon?
Not yet.
So this was a bear. It was found over on the east front of the Rockies here in Montana. So it weighed 400 kilos, was 2.3 meters high. And you may have read recently that there was somebody killed in Yellowstone by a grizzly bear. So I love this quote down here that Yellowstone Rangers advise parkgoers to stay far away from grizzly bears. But I think Gordon would tell you the same. So on Thursday, stay far away from grizzly bears. And the reason I'm showing this is because when this bear when this man was killed, the way they removed the bear from the park, but the way they identify who the right bear was is they took a genetic sample of the near where the man was killed, and then they captured a bear, and they looked at microsatellites to show that the bear that they captured was indeed the bear that killed them. So this is used routinely both in Glacier and Yellowstone National Park. So the main thing we're going to be talking about is the expanding role of genetics in conservation. And I like to think of, so I've been around for a long time, but I think initially conservation genetics was really dealing with captive breeding. So this is a tiger species survival plan, the American Association of Zoological Parks and Aquaria. And to me, what's really been the most exciting part is not using genetics to deal with captive breeding. I'm not a big fan of captive breeding, but to try to deal with wild populations. And I always like to give Charles Darwin credit for being the first conservation geneticist. And so Darwin, in this book, most people are aware of the origin of species, but if you're interested in genetics, this is really the book you should be reading. So Darwin talked about variation of plants and animals under domestication, where he talked about how traits are transmitted. And this was a chapter with a great title of Good Effects of Crossing and the Evil Effects of Close Interbreeding. So how many people here have ever written a scientific paper with the word evil? Well, maybe Robin, but not even Robin. So this was the chapter talking about inbreeding, and Darwin had a fairly modern view of inbreeding depression, to quote Gordon, but back then they called it interbreeding rather than inbreeding. And the reason that I say he's the first conservation geneticist, so people knew about inbreeding depression, but Darwin took that knowledge and thought about wild populations, and so he talked about fallow deer in British parks, and was worried about what would happen to these parks when habitat was lost. These deer were in these isolated parks, so they were completely isolated, small population size. He realized that there had to be inbreeding going on, so he went out and talked to the managers and found out that they moved animals around to get out of the problems, to infuse new blood from other parks. So I really think that was the first example of at least talking about genetics in terms of conservation. And today we would call that genetic rescue. And I'm a big fan of bumper stickers. So this is a bumper sticker I actually saw in Yellowstone National Park. Discourage inbreeding and ban country music. And I like it because first it's great, discourage inbreeding, but also I like all these little figures. So we've done a lot of work in the lab over the years looking at the effects of inbreeding depression on developmental abnormalities. These little guys here. And one of the rules in the lab was there's no country music. Like this. So, and if we bring that up to date, so the problem that Darwin talked about, the fallow deer back over 100 years ago, so most of the large mammals around the world today are in the same predicament. So 70% of the wild tigers are now found in 6% of the current distribution, which is less than 1% of the historical range. So there's many small isolated populations of tigers, just like Darwin talked about with fallow deer. And this was a paper that we published last year looking at how much gene flow is needed to bring about genetic rescue so that these small populations have some chance of survival to avoid the problems of inbreeding depression. And this is a picture my daughter took in Nepal last year. So the Nepal Tiger Genome Project. So again, everybody's trying to use genomics to try to assist efforts in conservation. So this probably should take more than 25 years now, but I think conservation genetics really began about 30 years ago. And there's lots of different applications. As I said, it started with captive populations, trying to describe relationships, kin relationships among individuals, look at the effects of fragmentation and isolation, hybridization, adaptation, just trying to understand the basic biology. So Gordon talked about counting grizzly bears in Glacier National Park using genetics, forensics, trying to identify bears that were involved in killing people. And eventually, I think in some cases, you could argue that genetic engineering, when it comes to species like trees, may be a viable way to try to assist efforts in conservation. So in Montana, and I think much of the world, genetics has really played an important role. So Robin Winkle, sitting in the back of the room, has been a primary biologist for NOAA fisheries, dealing with issues of the Endangered Species Act and salmon, largely using genetic data, trying to identify conservation units of salmon. So here in Montana, wolves have been very contentious. So wolves have been out here in the west, have been off and on the endangered species list several times. This really isn't a bumper sticker, but it's close to the back of the pickup truck. So this says Montana Wolf Management Team. This is the site. So wolves have been really contentious here in Montana. And so, as I said, they've been off and on the endangered species list. And most of the arguments, many of the arguments have been about genetics. And so this was the most recent case when they were put back on the endangered species list. And the reason was because genetic exchange is not taking place. So there were 3 different groups of wolves in the Pacific Northwest, well, in the Rocky Mountain West, on the endangered species list. And the Fish and Wildlife Service didn't deal with the importance of genetic exchange among those different populations. So genetics have played a really important role in trying to determine whether or not things should be protected under the Endangered Species Act and how they should be managed. So this is So this is a video taken by Jeff Stetz, the U.S. Fish and Wildlife Service. So these are wolves up in the North Fork, not too far from here. So you can see most of the wolves are black, and there's one gray wolf back here. So most of the gray wolves in Glacier Park are actually this melanistic. There's a couple other videos taken by Jeff, who worked with Kate Kendall, who Gordon talked about previously. So this is Robin. So this is a quote taken from Robin. So as Robin said, one of the times when I couldn't make it to the meeting, Robin gave this talk. And he talked about, as Gordon said, with the pipeline of data, how do we deal with all this information explosion. And this is a paper that Robin wrote that was published last year. So we sort of, with all the data, and one of the main points that I want to make is the importance of understanding basic concepts of basic population genetics. So Robin wrote this paper, published in the Journal of Heredity, and this was his motivation. I'm constantly appalled to see people go through the motions performing Hardy-Weinberg and lipid disequilibrium tests, recording the results, and then completely ignoring them, even when significant deviations are found. So I guess my main point is it's easy to generate a lot of data today, but really understanding the underlying population genetics is really important to understand those data. And this is a really nice paper. What could be more fundamental in population genetics than testing for Hardy-Weinberg proportions? As Robin talks about, there are a lot of problems in the literature of people doing that. So here's some data Gordon has been talking about filtering. So these are some data that Gordon and his group has produced. So FIS is the proportional excess or deficit of homozygotes. And when you look at 14,000 loci, you get a wide range of FIS values. So if whole populations in Hardy-Weinberg proportions, you expect the FIS value to be 0. So there's lots of loci which have very high FIS of 1, which means there's no heterozygotes. And there's lots of loci which have FIS very high negative values, that means are almost all heterozygotes. So what do you do with these data? How do you filter data like this? And as Gordon mentioned, Jim Sieb and Mike Miller are both going to talk about the problems of filtering data. And this is a small-scale example from a former student of mine who was working on kiwi in New Zealand. So Christina went out and looked at 30 microsatellite loci. So these are all the loci she looked at. And then she sent me an email saying, well, this locus here, there's a really high excess of heterozygotes. So I saw many more, 30% more heterozygotes than I expected. The FIS is minus 0.3, so I think we should throw this locus out. said to Christina, well, you know, there are good reasons why there might be an excess of heterozygotes. So, for example, have you looked at males versus females? So Christina went back and looked at the data, and it turned out that she saw only all the females were heterozygotes, and then most of the males were homozygotes for 178, 178. So birds have the ZW sex-determinant system. So what was going on was that there are two different alleles on the Z and the W chromosome, so that all the females are heterozygotes. And then when you combine them together, you get what looks like, well, it is an excess of heterozygotes, but the reason is because they're on sex chromosomes. So throwing data out, you may be throwing out the most interesting result that you have just because they don't conform to what you think the value should be. And so this is Roe, the species that she was working on. And Helen Taylor, a grad student in New Zealand, is looking at a little spotted kiwi, found the same result, different alleles, but the same result. So all the females were heterozygotes, again, because the two different sex chromosomes have different alleles, the 176 and 182. And all of the W chromosomes are fixed, or the Z are fixed for 182 allele. So all the males are homozygotes, and all the females are heterozygotes. So you have to be really careful in filtering data because you may be throwing out really interesting information. So, you know, I tried to figure out where I should throw this slide in. I wasn't sure where, but it's such a great slide. I had to use it. So, as everybody here knows now, you can do genetic analysis on almost anything, even a single cell. So this is when I was in Perth in Western Australia. So this is a sign which is actually on the buses in Perth. It says spitting is unacceptable. Drivers are now equipped with DNA kits to help with apprehension of offenders. So you can imagine what's the driver going to do? Go back and collect everything on the bus and take a genetic sample from everybody? So at least he must think it's going to scare people using that. So Gordon talked about Kate Kendall and her study. So these are the major two grizzly bear populations in the lower 48 states for Yellowstone and up here in Glacier in this region. And as far as we can tell, there's no exchange between them. So Kate set out to estimate how many grizzly bears are there in this region up here, what's called the northern the vine ecosystem, which is connected to them. So the goal of some people was to estimate the number of bears so that the argument could be made that they could be removed from the endangered species list. It was actually a very conservative Republican senator who came up with a lot of money so Kate could do this study, primarily so they could come up with a good estimate of the number of bears. So this is Kate. We'll meet on Thursday when she takes us on a hike. So this was during the 2008 election. John McCain kept talking about how millions of dollars were being wasted looking at genetics of grizzly bears. But as I said, the motivation really was from a Republican senator because the goal was to provide the data so the grizzly bear could be removed from the endangered species list. And it's really interesting. So there are two different ways. So they're looking at genotyping hair, looking at microsatellites. So there's two ways that they sampled hair. And Gordon, were there still any barbed wire on the trees on the Stanton Lake Trail when you were there?
No, and I think they had to take down the barbed wire everywhere.
Well, they just keep it up for a short time. So one way is bears rub against trees, and so you can see a chunk of hair where the bear rubbed. And they're worried about some kind of bias in sampling. So the other way they sampled was by putting out chunks of wood here and then pouring rotten fish nuts on top of the wood. So we track the bears. And then you see this wire around here on the bears. So under that wire, to get at the wood, they could get a sample. And a couple of videos, again, provided by Jeff Stetz. And this first video is, it's really not Gordon and embarrassing, but it looks it really does look like somebody in a bear suit. So what they do is they put barbed wire. You can tell which trees they like to rub against. They put barbed wire in the bear rubs. hair sample. So the other one I mentioned is the wood pile. So this is a grizzly bear. So you can smell the wonderful smell here of the rock fish cuts. So here's the barbed wire around it. The rock holding down the barbed wire. So the bear tries to get inside. When the bear goes inside, it leaves a hair sample in the barbed wire. So, trying to estimate the grizzly bears in the northern continental wild ecosystem. These were the 7,500 sampling locations. So you can imagine why the study cost a couple of $1 million. So we're sitting right here. So this is Missoula, which is 50, 60 miles, 100 kilometers south of here. Here's the Canadian border, because we know everything stops at the Canadian border. So these are either one of those two kinds of sampling, either a barbed wire or a tree, or there's other kinds of sampling. And then this is the results that Kate and Jeff published. So looking at the genotype the bears at 6 microsatellite loci, so they could tell individual genotypes how many different bears were they were collecting samples from. We're doing a capture-recapture study and using a wide-length marker that could tell whether they were male or female. So they got an estimate of 294 males, 470 females, total of 765 bears. And one of the important things under the Endangered Species Act was that you had to use the best available science. In the U.S., the courts have said if you're doing science, you have to have confidence interval on your estimate. So the previous estimates had been done by park people, and they flew over and they looked at the number of bears, and they just sort of they estimated how many bears there were just by looking at them, but they had no way of knowing what their error was. So the main reason that this study was done because they could estimate the error rate, and so the upper and lower bounds were 750 and 830. This was about double the previous estimates just based on observations. So Robin made another Robin slide talking about statistical analysis of the data. So one of the reasons everybody is here is how do we analyze these data that we're collecting? And as Gordon is talking about Tiago and others, using computers, modern computing power. And so it's a little bit humbling when you think of Sewell Wright. So Sewell Wright probably contributed more to population genetics than anybody else. And this is what Sewell Wright used for his computer. And this was a quote from Jim Crow. So Wright retired from the University of Chicago. His retirement is at the University of Wisconsin. And he sat in his office using his Monroe calculator, publishing all these wonderful papers. And I really like this quote. Jansky, so Jansky was an empirical population geneticist, and he published a paper with Wright. And I think a lot of us have these problems. So there are, if Jim Siebert here, Jim talks about theoretical dendrons. So these really high-powered theoretical population geneticists, and it's really hard to read their mathematics. And so this was Jansky talking about how he read the Sewell Wright papers. That my way of reading his papers is look at the biological assumptions and then skip to the conclusions and forget that and assume they're all met in between. And I think, certainly I read a lot of it as well. And so Jansky is Wright using the Monroe calculator. It used to be pretty hard even to do simple statistics, and now we have all these wonderful computers that do anything. So this is an email I got from Ayala, Francisco Ayala, who did his talk once. And Jansky had a technician and a half that did nothing but using a Wang calculator to calculate things like means and standard deviations. So it's just impossible to believe where we are today compared to where we were 50 years ago. So when I started graduate school in 1971, working with Joe Felsenstein, Joe had one of the first Hewlett-Packard calculators. So back then, even calculators were around, let alone computers like we have today. So how do we use all this information? So Robin, there's a lot of cautions. New methods aren't infallible, which Mike Miller and others are going to talk about. So just because we have programs doesn't mean that they're going to provide all the answers. We really should be skeptical, as Mike Miller. And I guess my main point is this: that understanding theory is crucial for using this power. So you can collect all the data in the world that you want, but if you can't understand it, then the data is not doing you any good. So it's like having a book, which is this wonderfully written book. If you can't read the language, it doesn't do you any good. You can't understand it. And so this is what I think one of the major purposes of this course is to get people so that they begin to apply and understand population genetics theory, analyze all these data. So this is my stool. So making the point that going out and collecting data, we really have to be able to do three different major things. So we have to collect the data, we have to be able to analyze the data, but also we have to understand the theory to interpret those data. And to really do a good job of empirical population genetics, we have to be confident in each one of these three areas. And I've been reading this book by Svante Pabo on trying his efforts and others working with him, trying to look at and come up with a genome sequence of Neanderthals. And I was reading this book last night, and I came across this quote, which I really love. First and foremost, we needed population geneticists. So they had all these data, they had all these sequences from Neanderthals, but they didn't know what to do with them because they didn't have a population geneticist to help interpret. So he talks about contacting with Richard, this week, Mike Slack, and others, but without having population geneticists, they couldn't interpret these data. And so I think everybody in here, if you really want to go out and collect next-gen data, if you want to end up being more than a technician, you really have to understand population genetics. And I have a couple of books to recommend to you. So the first one is Crow and Kimura, which was published in 1970. And it's an old book, but the theory is still really valuable. And this book was a couple of years ago, it cost $100. You can now order it from Amazon for $50. So I like to tell people, everybody who considers himself a population geneticist should have this book on the shelf. They should look at it. So Robin's going to talk about effective population size. Well, if you really want to go back and see where did all this theory come from, Crow and Kimura is a way to start trying to understand theory. So if you can't afford $50, so this is Joe Felsenstein's Notes in Theoretical Evolutionary Genetics, which is population genetics. So if you Google Joe at the University of Washington, you can download this for free. It's online. That's another great source. And this is obviously, so this is up to date every year that Joe teaches it, and the notes are updated. So and Joe is he may have been Jim's original theoretical minimum. But Joe really does a good job of trying to explain things in words that those of us who are not mathematically gifted as Joe. So the third I would recommend is the book that Gordon and I wrote. And so in our motivation was trying to, what I'm saying now is trying to connect, looking at empirical data, but also trying to present the theory. And when we present the theory, we try to present examples. And so for years, going way back to the beginning of genetics in 1900, in terms of the empirical work for 70 years, it was basically laboratory matings and chromosomes. So there really wasn't population genetics in the wild. Then in the 1970s, Gordon talked about the Dick Lewontin, allozymes, protein electrophoresis came along, 1980s, people dealing with mitochondrial DNA, 1990s, microsatellites and other types of nuclear DNA, and now in 2000s, we can look at genomics. So there's been this amazing increase in our power to look at empirical population genetics. And so I like to think of this as 3 different years. So from 1900, 1920 to 1970. So we had Sewell Wright, we had J.B. Saltine, we had Arthur Fisher, who were all mathematical geniuses, who most of the theory we use today came out of those 3 brains between 1920 and 1970. So we had a lot of theory and models available, but we had very little empirical data, and there was very little analysis dependent on these handy calculators. So for the first 50 to 70 years, I think this is what population genetics looked like in terms of the 3 legs of the stool. And then I began graduate school in 1971. So this is the golden years. So now we're able, using allozymes, people are actually able to go out and look at genetic variation in any species, from E. coli to elephants. So now we can collect empirical data. Computers came along, so we had this balance among these 3 stools. But I think where we are today is now we have this fire hose of data. And it's not that we have less theory and models available, but relative to the amount of data that we have available, it's just a huge circle. And then we have lots of power available for analysis. So I think the key now is not generating data. As Marco Pavo said, they had lots of data on the Neanderthals. The problem was they couldn't interpret it without having really gifted population geneticists trying to understand what they were looking at. And so a few years ago, Paul Hohenlohe and Gordon and I wrote this paper trying to think about, so how is genomics going to change applying genetic issues to conservation? And it still is not clear, in my view, how much. I think there's a lot of hype about genomics, and genomics is really exciting. Understanding evolution, understanding the Neanderthals and everything, genomics is wonderful. But trying to apply it to conservation, it's not clear how, what can we, so my question is, what can we do now that we couldn't do before when we had genetics? And that's one of the issues we tried to address here. And so this was a box that we came up with looking at. The blue boxes are what we could do traditionally with satellites or allozymes. So when you look at, we could estimate population size of grizzly bears. We can look at genetic drift, we can look at inbreeding, we can look at genetic diversity, but it really wasn't until we could start to do genomics that we could begin to do things like estimate migration rates, begin to look at local adaptation, begin to understand outbreeding depression, begin to understand inbreeding depression, as the papers that we read today in my talk about. So Gordon mentioned this paper, which, as he said, is largely a bunch of European authors, and Mike Schwartz buried somewhere here in the middle. And so one of the points, the main point of this paper, is that conservation operates in this circle, real-world conservation issues. Then we have academia, people like many of the people in this room who are publishing things. But there is not the point of the paper was there's not much interaction between the academic publications and real-world conservation issues. And I think there's truth in this, but I think it's more true in Europe. Mike was the only non-European author. It's more true in Europe than it is in North America, because we're lucky enough that people like Robin Wakeles working for the U.S. government, or fisheries, we're lucky to have Mike Schwartz working for the U.S. Forest Service. So I think in the U.S. there are more really good population geneticists who actually work in these management agencies. But so I think this largely this idea that these are separate is largely European influence, and it's not so true in the US. So as Gordon mentioned, he and his students have written a response to this paper. This is published in Trends in Ecology and Evolution. And so there's many people in this room who are co-authors here. And Gordon's here, Lisa's here, Jim is supposed to be here.
It's in review. There's still time for.
Steve O'Brien, so Gordon did mention his nickname is SOB. John Winberg, well, John's not here. So, and one of the points they make in this paper, and I think it's really important, is that genomics is used very loosely. And it's used, you can either think about in the broad sense or the narrow sense. So in the broad sense, this is a quote from the paper, it's the use of new genomic techniques and genome-wide information to solve problems of conservation. The narrow sense definition extends further, requiring the use of qualitatively or conceptually different approaches. And so, in the broad sense, genomics is just using more loci becomes genomics. But I think in the narrow sense, genomics is conceptually different. It's not just doing what we did before, having more loci, but it's really using things which are conceptually different. And I really agree with this point, although I think they misrepresented it in the paper, because this is one of the tables, and in there, so they talk about examples of genomics and conservation, and then they say whether they're narrow sense or broad sense. So this one is what they say to be looking at Atlantic salmon. They say using genomics, and they say it's the narrow sense because they use outlier tests. Remember, the criterion for a narrow sense is something conceptually different. And so outlier tests were developed in 1973. So what, 27, so 52 years ago, people were already doing outlier tests. So I don't think outlier tests are conceptually different. But there are areas in conservation that are conceptually different. So I think one of the things that would be worthwhile for you to think about is what we're doing, is it just looking at more loci, or are we doing something which is truly genomic and really conceptually different? And for example, here is a paper, so this is published in 2001, so I don't mean to beat up on these people, but it's a nice example. So they talk about population genomics, and one of their examples of genomics approach is 13 Alzheimer's. So I think most people in here would agree that 13 loci in mitochondrial DNA is really not doing genomics. So you have to do more than just look at loci, more loci. And an example of there is more power when you look at more loci. So this was a paper looking at humans. And these are different human groups, about 20 different groups, looking at SNPs. And this is looking at principal component analysis. So if you look at 10 SNPs, you see sort of a shotgun. There's not much signal there. Or if you look at 100 SNPs, things become more clear. And you begin to see that these different groups are more colorblind. I'll tell you what the groups are. But you do see the colors cluster together. And if you go from 10 to 100 to 1,000 SNPs, now you see these different groups. So we're looking at more loci, but it's not really conceptually different. It's just looking at more of the same, but we get more power by looking at more loci. But I think it's more interesting to think about, well, what can we do which is really conceptually different? And I think one of the best examples is here, inbreeding depression. So really trying to understand inbreeding depression in a way that we couldn't before. And this is, I think, from my view, this is the most exciting application of genomics to conservation. So this is a paper that Marty Cardos, who's a student of Gordon and I, published in Heredity this year. So traditionally, people have looked at pedigrees and looked at F, the inbreeding coefficient, which is the expected reduction in heterozygosity due to a common ancestor in the pedigree. So should we use pedigree F? And it's really hard to generate pedigrees. And this is what people have done now for over 100 years. But now that we can look at genomics, are we better off using pedigree F, or are we better off using genomics? And the answer is forget pedigree F, we should be using genomics. And so this is Marty's explanation of that. So historically we've used pedigree F, which is the expected proportion of the genome that is identical by descent because of having a common ancestor. So you inherit the same chromosomal segment from your mother and your father that were in a common ancestor. Therefore, you have reduced heterozygosity. The expected reduction in your heterozygosity is your F value, the inbreeding coefficient. However, your F value does not capture variation among individuals with the same pedigree. And I'll show you in the next slide that can be considerable. And the other thing is, when we start with a pedigree, we assume that this is the beginning of time, and there could be inbreeding which occurred before that time. So your pedigree is, you can't take into account that there was inbreeding before the time that you begin looking at the pedigree. But looking at the variation in F, so this is really, Ian Franklin published on this a long time ago, but it's something that people really didn't pay attention to. So these are full sibs. So the expected F value is 0.25. So you expect a 25% reduction in heterozygosity due to inbreeding, but depending on the size of the genome, if you have a small genome, there's a much greater variability. If you have a large genome comparable to humans, there's still substantial variation. So these are the actual proportion of the genome identical by descent. So even though we expect 0.25, the actual values go down almost to 0.1 up to 0.35. So trying to detect inbreeding depression, F is such a noisy indicator of proportion of the genome identical by descent, it's not surprising it's been difficult to detect inbreeding depression in many species, relying upon the pedigree. And so what should we do if we can't use the pedigree? And so what we're going to use is what people developed with humans about 10 years ago, looking at runs of homozygosity. So here we have a pedigree. Here we have, and we're looking at this individual here. So this individual, his grandfather was the father of both his father and his mother. So it's possible that part of his genome will be identical by descent because he could inherit the same chromosomal region through both his father and his mother because of the common ancestry. So just looking at Mendelian transmission, so this individual gets one chromosome from this parent, one from this. There are crossovers that take place, so rather than getting the whole chromosome, you get chunks of chromosomes. And the mother, and then we have this individual. So this individual happens to inherit these chromosomal segments that, if I wasn't colorblind, I could trace back to the three parents. What we're really interested in are regions that came from this individual, and these are so-called runs of homozygosity. So here we have that chromosome. So in this region right here, the individual inherited the same chromosomal region from his mother and his father because of the grandfather being the parent of both. And so this is the proportion of the genome identical by descent. These chromosome tracks because of the fact of the common ancestry. So we can estimate the proportion of the genome identical by descent by looking at how large are these tracks over the total genome size. And so here's an example. So this is Marty's now working as a postdoc in Hans Ellegren's lab. So these are some data looking at one chromosome in Swedish flyer called flycatcher. So this is the chromosome from 0 megacentomorgans out to, well, this is about 70, almost 80 megabases. And so this is heterozygosity at SNPs, and you can see these regions where, you can see this region here where the heterozygosity is greatly depressed, and this is a region which is identical by descent. So these are regions which are not identical by descent. So by looking at the regions which are identical by descent, we can come up with a much better estimate of the total proportion of the genome, which is identical by descent, because by looking at these kinds of data. And the other thing is, not only can we estimate the proportion of the genome, but also by looking at how long these sections are, we can tell whether the inbreeding occurred last generation or many generations ago. So if the inbreeding is recent, then we would have big chunks like this. However, if the inbreeding occurred many generations ago, so there's been lots of recombination, we'll have lots of small chunks. So not only can we estimate the proportion of the genome, but we can also get an idea how long ago the inbreeding occurred. And so this is a paper published this year looking at woolly mammoths. And this is really science fiction stuff. So looking at, woolly mammoths went extinct about 11,000 years ago from the mainland of Asia and North America. But there were some woolly mammoths were left. This is Wrangel Island. On these islands, they persisted for a long time. So they had 2 samples of woolly mammoths, one from the mainland and one from Wrangel Island. So just 2 samples. And it was about 44,000 years ago, the mainland sample, and estimated to be about 4,000 years ago from the Wrangel Island. And so their question was, were woolly mammoths on Wrangel Island, did they have less genetic variation because they were small and isolated? And they also were interested in looking at the demography. So as the title says, looking at the demographic and genetic declines. So using a coalescent analysis, which I'm sure most people here have heard about, you're going to hear a lot more about it. By looking at these 2 separate samples, 30,000 years apart, they estimated the population size of woolly mammoths scaled to 4 times the mutation rate times the effective population size. And so they got this demographic history, which is, to me, the most interesting thing is that how the 2 separate samples came up with a very similar change in population size over time. So by looking at the coalescent, looking at these genome data, this is something we couldn't do before. You can estimate the demographic history of the population. And more importantly, so this is right before it went extinct. More importantly, so this is heterozygosity over the genome. So these are from orangs, which have really high heterozygosity, down to snow leopards and Tasmanian devils, which have really low heterozygosity. Looking at the whole genome. But the key thing here is looking at the 2 woolly mammoths that we were talking about. So the Wrangel Island one, it doesn't look very dramatic, but it's about a 20% less heterozygosity. So apparently there's about a 20%, if we assume that these at one point had the same heterozygosity when Wrangel Island was connected to the mainland, there's been a 20% reduction in heterozygosity. We can't tell when that occurred, but looking at runs of homozygosity, so these are just 2 chromosomes. So here's the mainland population, and then looking at the Wrangel Island, we can see all these bars up here represent tracts of the chromosome which are identical by descent, which represent runs of homozygosity. So on these 2 different chromosomes. So in the Wrangel Island, there's about 23% runs of homozygosity and less than 1% in the continent. So again, it's about a 20% reduction in heterozygosity. But now with genomic data, we can say sample size of 1 is not very impressive. But we can say that this loss of heterozygosity is not because this individual is his parents, recent inbreeding, and Nielsen call lengths of migrant tracts. So rather than looking at homozygosity, we're looking at tracks where you can see exchange of chromosomes coming from two different populations. So by doing that, we can estimate how much migration there's been between two populations, and we can estimate how long ago this occurred. And I think one of the nice examples of this is a paper published a couple of years ago. So looking at, so I think most people here know that there's now evidence that everybody in this room, about 3% of the genes in this room came from Neanderthals. But the question is, when did that admixture, when did that hybridization occur between modern humans and Neanderthals? And by looking at the lengths of these chromosome tracks, these two papers, they were able to estimate when, and again, David Reich is one of those population geneticists, Swampy Papa said he really needs to analyze his data. So by looking at the lengths of these tracks, you get an idea how long ago this admixture occurred. And apparently it was something that occurred when modern humans first came out of Africa. Apparently right after they came out of Africa, there was this admixture hybridization. So this is what I really think of as population genomics or conservation genomics. So we're doing things that we couldn't do before. And I just have a couple of one-slide other examples. So this is work called Hohenlohe and Susie Bassam, looking at sticklebacks, three-spine sticklebacks. So this is an outlier test, but it's what I call a genomic outlier test. So here we have a chromosome. So now these SNPs are put onto a chromosome map, and we can look at the FST values, and we can see there are these loci which are outliers. So the average FST is about 0.1. And this is between marine and freshwater sticklebacks in Alaska. And there are these regions which are much higher, and those are, I didn't want to show more than one slide, but they've looked at a number of these independent populations derived from the ocean, and you see the same regions are outliers, and by QTL analysis, they know which loci are found in these outlier regions. So rather than just looking at a locus and say it's an outlier, this is allowing us to begin to get at what is the functional basis. So this is other work done by Marty Cardos. So Marty did a bunch of genomic work with newborn sheep, and similar to the work, analogous to what Paul Hohenlohe did. So looking at the heterozygosity value over a chromosomal region, there is this region, so the average heterozygosity is somewhere up here, and there's this region where heterozygosity drops way down. We blow that up and we'll see that. And so this region looks like it's been a selective sweep. So for some reason this region was under strong positive selection and increased in frequency, and by doing that, this great increase in frequency, it dragged along the rest of the chromosome, so a lot of the heterozygosity was lost. So due to a selective sweep and hitchhiking effect, we can find these regions of the genome. And it turns out that this is a gene which is associated with large horn size in domestic sheep. And so at least the inference is that this is a locus which was associated with a lot of bighorn sheep. Big horns because of this selection. So I'm happy to answer any questions if there's some. Thanks. Your hand is up first.
I was just wondering if it's easy to differentiate between the loss of heterozygosity from the hitchhiking effect of selection and inbreeding effects of predicting patterns.
Well, the difference is that inbreeding should affect more than just one region. So if you see that effect only at one region, that would suggest that it's selection. So like in the mammoths, if you see a whole bunch of regions where you see those ones of homozygosity, that would suggest that it's inbreeding. So inbreeding is a genome-wide effect, and selection is a locus-specific effect. So,
it's So that question I have about the present on the inference of historical change in the migration in relation to the length of the track. Like I was wondering, how do you do with the impact of selection on the lower recognition rates and the hotspot of divergence in the genome and how it's possible to discriminate these things? And when you talk about time, is it accumulative lengths of all the genomes? So you consider all the genome instead of just number of tracks in the genome?
Well, so you begin, so looking at those tracks,
Yeah.
you begin assuming that recombination rates are uniform over the
Yeah,
genome,
so you assume
which is not
it's
true.
not
Which is not true. So that's sort of, you have to begin with simple models. But then it becomes, but I think in the broad picture, that's what you're looking for. But then there are regions that there really are, like you said, hotspots of recombination. And so it does become much more complicated.
And in this method, I didn't read the paper, I will for sure, but in this method, do they remove all the diversions?
Well, the Neanderthal paper is really hard to get through. I mean, the data are just overwhelming. So I really think, the book, Pablo Svante's book is really great. And I've been reading that for a couple of weeks. And it's really, what I like about it is he goes through and talks about analyzing data. So finding regions where there are differences between modern humans and Neanderthals, how do you determine whether those are errors, whether those are contamination, or whether they are a real biological state? And it's really, really, really complicated. And I think if you read that book, you'll get some idea of some of the complications. But it's a great book. And plus, you find out more about other things than you would ever imagine you're going to find out, as well as this. Especially when he describes his wedding ceremony on the big island of Hawaii.
Any more questions?
So do you think we'll start seeing the pendulum kind of swing back the other way to using fewer markers if we don't need so many markers? If we're not putting that
Well, I remember, so I think it's a really important, when you're going to go out and collect data, it's really important to think, how much data do I need to answer the question? And I think there's a tendency, I'm going to go out and do genomics. And if you want to get a job, that may be a good motivation. But if you're really interested in answering a conservation question, I think it's more important to ask, okay, how many markers do I really need to answer that? Or sample size. And I think those two things, I think it's really easy just to go out and collect data, but it's really important to sit down and think, what is the question and what information do I need to answer that question? Sometimes we need lots of markers, and sometimes we don't. I know that Jim and Lisa have done work looking at Chinook salmon in Alaska, where looking at allozymes and microsatellites, there's sort of a hint of some population subdivision, but it wasn't until they looked at many SNPs that they could really see that subdivision. So I don't think there's no answer in how many loci, how many markers one need. Depends upon your question. And how differentiated the populations are.